Today, let us start with biochemistry first lesson, biomolecules and the cell. 90% of the dry weight of body is made up of carbon, hydrogen, oxygen, nitrogen, phosphorus and sulfur. So these are all chemicals. So they're saying 90% of body is made up of chemicals. Now let us study about all these biochemicals in this biochemistry. Now several elements like calcium, magnesium, sodium, chlorine, potassium, uh, molybdenum, etc., and are also present in our body, which are helpful in our body biological functions. Now, carbon is most predominant and has unique property of forming C and C single bond, which forms C and C chains of unlimited length. So, 90% of compounds found in system are carbon. And now life is full of lifeless chemical molecules like for example, single E. coli molecule has 6,000 different organic compounds, and man may have about 1 lakh types of molecules. Here we can see the table. The complex biomolecules are made up of monomeric units like for example, here we took 5 types of molecules. First is protein, where protein is made up of monomeric units of amino acids. Then it is the function of it is fundamental structure. It gives fundamental structure to cell. Then deoxyribonucleic acid or DNA is made up of deoxyribonucleotides, where it helps in repository of hereditary information. Then ribonucleic acid or RNA made up of ribonucleotides, which is essentially required for protein synthesis. Now polysaccharides, or it is also known as glycogen, which is made up of monosaccharides or glucose, which is energy form, which gives energy to the body to meet short term needs. Then lipid uh, is made up of monomeric units such as fatty acids and glycerol, where it is a source of energy form which helps in meeting the long term needs, and it also gives structural component. It also makes most of the structural component of membranes. Now let us see the chemical composition of man of 65 kg. Here water is in abundant state, that is 61.6% or 40 kg. Then protein 17% or 11 kg. Then lipid 13.8% or 9 kg. Carbohydrate 1.5% or 1 kg. Then minerals are 6.1% or 4 kg. Now here you can see the cell. Cell is a basic unit of biological activity, where it was first seen by Skleden and Swan in 1838, but the complexity and complex structure was given after 1940. And we have two types of cell, mainly prokaryotic and eukaryotic. Now let us see the difference between prokaryotic as well as eukaryotic cells. Character is first is size. Here the prokaryotic size is small, that is 1 to 10 micrometer, whereas eukaryotic cells have large size, that is 10 to 100 micrometer. Then cell membrane, prokaryotic cells have cell wall, whereas eukaryotic cells have plasma membrane. Now subcellular organelles are absent in prokaryotic cells, whereas they are present in eukaryotic cells such as mitochondria, Golgi apparatus, etc. Then nucleus is non-defined. DNA is found as nucleoid. Histones are absent, which are in prokaryotic cells, whereas the DNA is defined and covered with double membrane and associated with histone proteins. Now energy metabolism, there is no mitochondria present in prokaryotic cells, and enzymes of energy metabolism are bound to membrane only. And here the eukaryotic cells, the enzyme of energy metabolism are in the mitochondria, which is present in the eukaryotic cells. Now cell division does not usually occur in prokaryotic cells, so usually fission will occur, and in eukaryotic cell mitosis will take place. Then cytoplasm, the organelles and cytoskeleton are absent in the prokaryotic cells, whereas the organelles as well as cytoskeleton is present in the eukaryotic cell. So eukaryotic cells have a definite structure when compared to the prokaryotic cells. We have 250 types of cells in our body which are eukaryotic, and 10 to the power of 14 cells of eukaryotic cells are present inside our body. Now let us move on to the first cell organelle, nucleus. Nucleus is the largest cell organelle, and it is double membraned. As you can see here, it is double membrane, and the inner membrane and outer membrane, the distance between them is 90 nanometer, and the outer membrane is plain and continuous with endoplasmic reticulum, whereas the inner membrane has pores which are known as nuclear pores, which allow the material from nucleus to cytoplasm or from cytoplasm to nucleus. It has as passage between both nucleus as well as cytoplasm. The nucleus has DNA associated with histone proteins, as we saw in eukaryotic cells, in the ratio of 1:1. Here, to form the chromosomes, the nucleosomes are thread-like structures which together form chromatin, and chromatin will together condense together to form chromosome. So we can say that single chromosome has millions of nucleosomes. In human body, we have 46 chromosomes packed in a nucleus. Here you can see this is structure of nucleus. So inside nucleus we have a dense body called as nucleolus, which is rich in RNA, particularly the ribosomal RNA, which enters the cytosol by nuclear pores which are present here, and these ribosomal RNA which help in protein synthesis. And nucleus also have nucleoplasm, which is fluid-like or gel-like substance present in nucleus, and which is rich in DNA and RNA polymerases. Now here you can see a syndrome named Hutchinson-Gilford progeria syndrome, where it is very rare with we can see one case in 5 million. It causes aging in children from the birth, and it is seen due to accumulation of abnormal protein called as lamina A in the nucleus. Now let us move on to second cell organelle, mitochondria. Here it is also, as we know, mitochondria is also known as powerhouse of cell, and usually the size of mitochondria is 1 × 3 micrometer. About we have 2000 mitochondria occupying our cell, up to 1/5 of the total cell volume, and the mitochondria is double membraned. As you can see, the inner membrane are forming inner foldings known as cristae, and the matrix is also known as mitosol, which is rich in double-stranded DNA, RNA, and ribosomes. Now the third cell organelle is also known as endoplasmic reticulum, which is divided into two types: smooth endoplasmic reticulum and rough endoplasmic reticulum. The smooth endoplasmic reticulum helps in lipid synthesis and supply calcium ions for cellular function, and the rough endoplasmic reticulum contains ribosomes like nodules like substance, so they are called as rough. Then these help in protein synthesis. These endoplasmic reticulum are extended from the nuclear pores which are present here. As you can see on the nucleus pores are present. From nuclear pores to the plasma membrane or cell membrane, they are extended from there to there. And during the division of rough endoplasmic reticulum, they form small vesicle like substances. They are known as microsomes, and which do not occur in cells. Only during division they occur. Now Golgi apparatus, it is the structure is similar to endoplasmic reticulum, but the function is different. Here the endoplasmic reticulum, after synthesis of protein, send this to Golgi apparatus. So here the work of Golgi apparatus is addition of carbohydrates or lipids or sulfate moieties to the newly sent proteins from the endoplasmic reticulum, and it helps in membrane synthesis for formation of intercellular organelles like peroxisomes, lysosomes, etc. Lysosomes. Lysosomes are spherical in shape, which are single layer, and which are also called as digestive tract of cell. They help in digestion protein, lipids, carbohydrates, and nucleic acids. and they are categorized into hydrolases. For example, alpha glucohydrase, which synthesis glycogen, then cathepins synthesis proteins, then lipases lipids, ribonucleases RNA. In lysosome, lipids and proteins are remained as residuals, and they are called as lipofuscin, which are released when the cell dies, and they help in post-mortem autolysis. And some components escape from lysosome during their synthesis, and they release into cytosol, causing arthritis, muscle disease, allergic disorders, etc. Inclusion cell disease or eye disease is a rare disease due to absence of hydrolysis in the lysosome. Now peroxisomes. Peroxisomes are also spherical, oval, and they are single membrane. They are also called as microbodies, and it contains enzyme called as catalase. Catalase help in converting H2O2 to H2O and O2. And peroxisomes oxidize long chain fatty acids which are greater than C18, and the synthesis of plasmalogens and glycolipids. Plants have glycoxysomes, a special type of peroxisomes, which involve in glycoxylate pathway. Now, peroxisome biosynthesis disorders, PBS, example, Jewell's syndrome is a rare disease characterized by absence of functions of peroxisomes, and the child may die after birth, after 1 year of birth. Now, cytosol and cytoskeleton. The cellular matrix or gel-like substance in the cell or cytoplasm is also known as cytosol. In cytoplasm or cytosol, we have many enzymes, metabolites, and this gel-like medium or substance is seen. Now, cytoplasm has complex network like protein and filaments, which are called as cytoskeleton. Cytoskeleton filaments may be of three types: microtubule, actin filaments, and intermediate filaments. And filaments with polymers of protein are responsible for structure, shape, and organization of cell. So here, cytosol and cytoskeleton are main sources to keep the cell in shape and maintain the structure of shape. So here we have completed the first lesson of biochemistry. Thank you.